A warm greetings to one and all. And in this video, we are going to discuss about the functions of plasma membrane. Plasma membrane is a thin, elastic, semi-permeable membrane that serves as a boundary for the cytoplasm. And this term, plasma membrane, was coined by Nageli in 1855. This plasma membrane has an enormous functions in the living organism. So what those functions? So let us see one by one in this session. So first thing is this plasma membrane is going to provide the mechanical support. It is going to provide the mechanical support to give a definite shape to the cell. And next one is the transport of materials. So when we discuss about the transport, we need to talk about different types of transport like passive transport, active transport, osmosis. And in the beginning itself, I told that plasma membrane is a semi-permeable membrane. So what is the semi-permeable membrane? It does not allow all the molecules to get inside the cell. Whatever is required, what is necessary, only the necessary substances will be allowed to move inside the cell and move outside the cell. That normally we call as semi-permeable membrane. Okay, so in this passive transport, we doesn't spend more energy. That is, the molecules from the higher concentration moves to the lower concentration. This normally we call as downhill movement. So usually when we climb the hill only we need more energy. Whereas when we climb down or when we are coming down the stairs, we doesn't require more energy. The same way here also the molecules does not require any energy to move the materials inside or outside the cell. Okay. See in the image also shows you or illustrates you very clearly. So this a line serves as the semi-permeable membrane and this is lower concentration and this side is higher concentration. So from the higher concentration, the molecules move to the lower concentration. So this bicycle riding shows without spending energy, from the top hill we can come down easily. So this is called as the passive transport. The next one is the active transport. When we see about active transport, it requires more energy to transport the molecules to inside or outside the cell membrane. It needs more energy. That is ADP, energy currency, adenosine triphosphate molecules. That is, this is moving from lower concentration to higher concentration. This is uphill movement. When we are climbing the stairs or when we are climbing the mountain, we need more energy. See the same picture, but the perception varies here. Lower concentration, higher concentration, but the molecules are moving from lower concentration to higher concentration. See, the bicycle is moving up, so it requires more energy. Okay, and the next function is exocytosis. So what is this exocytosis? When the molecules or the cell is vomiting something outside, waste materials or the important secretions, anything it can be. Normally, we talk about the waste materials. After some process, the cell needs to send out the waste material. So that will be sent out in a process called as exocytosis. Okay, see here. You can see the image very clearly. The Golgi apparatus is there, and the Golgi apparatus forms the vesicles. It is the secretory vesicle, and here it might be the proteins or important materials synthesized by the Golgi apparatus, and that also needs to be sent out of the cell to reach particular target site. And here some sort of waste will be produced, and that needs to be sent out. Say, for an example, when we cook in the kitchen, some food will be prepared and it will be taken to the dining table. Okay? The same way, when we are cooking, some sort of vegetable waste or fruit waste or something else that need to be sent outside, or it needs to be thrown in the trash. So that is also done here. So this vesicle attaches with the plasma membrane and it secretes the waste material outside, releases their content. Hope this image gives you a clear idea. You see, this GIF image that also shows a wonderful explanation. So the phospholipids from the vesicle membranes. So this phospholipids from the vesicle membrane integrates with the plasma membrane. So we know that plasma membrane has the bilipid layer. So this is getting merges or integrates with the plasma membrane and the molecules are sent outside. So this is an example for exocytosis. So this is an example for semi-permeable membrane. It allows only certain molecules to cross. So what are all the molecules? Non-polar molecules like carbon dioxide, oxygen, and small polar molecules like water, larger polar molecule like glucose, and ions like sodium, potassium, etc. So this is outside the cell membrane and this is inside the cell membrane. It needs to be transferred inside. Okay. And this is the structure of the plasma membrane. This is the head region and this is the tail region. So it is the bilipid layer. You have two layers of lipid in between proteins and this is the protein, transport protein. They allows the exit or entry of certain charged molecules to enter the cell. So these proteins are very, very specific and only glucose can pass through the glucose transport protein. See, this protein is very specific. This protein is specific only for the glucose. So only glucose can be transported through this protein. So next one is transmembrane transport. Already we saw about this. And what is happening? See the integrins. I said in between we will be having the proteins. In between the bilipid layers, you will be having the proteins, that is integrins. So what this integrin is going to do? This is going to communicate or adhere the cell, adhesion process. Nearby cells will be adhere to one another with the help of this protein. See the cell and cell to cell and cell matrix adhesion. So this is one cell and this is another cell and these are getting adhered with the help of certain important structures, what we call as tight junction or occluding junction or zonular occludence. And here you have another type of protein called as zonular adherence, and this is called as desmosomes or macular adherence, and this is the gap junction. And here you have the hemidesmosomes, and in this region you have the focal adhesion. So in these points, the cell will be adhered to one another. And This is done with the help of the proteins, what we call as the integrins. So this is membrane transport. As we already said, many GIF images are there to make you understand in a very, very clear way. So when you see the picture as well as you hear the lecture, you can understand better. So this is very simple diffusion. The carbon dioxide and the oxygen is moving inside and outside the cell. So carbon dioxide moves outside the cell and oxygen enters the cell. So since it is the semi-permeable, it allows only the required one. And here doesn't require any energy. It is the passive transport. And next one is nerve conduction. So what is this nerve conduction? What it is going to do? The plasma membrane is going to conduct transmission of impulses. It is going to transmit the impulse. Depolarization and repolarization. Every time the cell needs to be depolarized and repolarized. Every fraction of second, this will be happening. So what happens during the depolarization? The sodium molecules are entering inside, and during the repolarization, the potassium ions are coming outside. See, in the cytosol is nothing but in the cytoplasm solution like substances, cytosol and cytogel. So it is a matrix which is present in the cell. So that matrix contains sodium and potassium ions, that is gated channels. This is going to operate the entire movement of the molecules. So what happens is during the depolarization, outside from the outside of the cell, the sodium molecules moves inside. This is getting depolarized. And when the sodium molecules or sodium ions are getting inside, in another stretch, what happens? The potassium ions are coming outside. That is repolarization. This is only conduction of impulse or transmission of impulse. And this is cell recognition. Very, very important thing. The cell needs to recognize, or the plasma membrane obviously need to recognize which is the favorable material or which is the foreign particles that needs to be identified. So some foreign bodies like bacteria or if something else enters inside the cell, the plasma membrane identifies or recognizes and engulf them by a process called phagocytosis. So it is giving an emergency alert. See, when the foreign body comes, so this is giving an red alert. Immediately, the plasma membrane engulfs. Okay, that is similarly the macrophages of the spleen can identify the worn out RBCs from the healthy RBC and destroy them. This is very similar to that. And this is cell signaling and communication. So the plasma membrane need to give the cell signal. So as I said already in this cell recognition, so it needs to give the signal if foreign bodies are identified or if the necessary materials are taken inside or it has to be sent out, that needs to be given a proper signal and communicated to rest of the organelles or to the nearby cells or to the organ system. That need to be communicated in a proper way, and that is done with the help of the plasma membrane. See here, the signal transduction pathway, and this is the plasma membrane. When the molecules, signal molecules, extracellular fluid, and here the molecules come and attach to the receptors, immediately it is giving the response. When it receives, when the receptor receives the molecules, automatically the transduction takes place and gives the signal. So this is done with the help of plasma membrane. So that needs to be done. It is very, very important. See the secretory vesicles, and these vesicles will be taking out the substances. And next, foremost and important function of plasma membrane is homeostasis. So what is this homeostasis? Our body needs to maintain the or regulate the temperature as well as the salt substances. Osmoregulation, that is very important. So when the temperature increases, we sweat, we lose more water, we get dehydrated. But activating the sweat gland, it is removing the heat, the way of the perspiration reduces the body heat to maintain our body temperature. At the same time, the water needs to be balanced in our body, and that is done. Water as well as the iron molecules. In the sweat, some amount of salt is also excreted, and that also needs to be balanced. And that is done with the help of this plasma membrane. And this is the antigen specificity. What is this antigen specificity? So the cells located on the surface of the plasma membrane specifies the antigen. The antigen determinants are the glycoproteins, and the rejection of transplanted tissues during the transplantation or grafting. Some organs will be grafted or transplanted, and that is rejected based on the determination of the antigens or the glycoproteins located on the cell membrane of the implanted cell. See, this is the new cell, and this is your original cell. And here, the endoplasmic reticulum or the Golgi apparatus, they will be sending the proteins, glycoproteins, and that proteins will be attached to the plasma membrane. When this, assume this as a grafting organ, and this will be producing different type of glycoprotein. So this will be rejected. Based on this glycoprotein, it will be rejected. If both the things are having the similar structure of glycoprotein, then it will be accepted. So for that only, before undergoing major surgeries for transplantation or for grafting, they will be compatibility. They will be doing the compatibility test, how the glycoprotein or the antigen is getting matched. After that only the organs will be transplanted. See, when the immune system detects the antigens, a structure will be produced, a specific shape that has to match, like lock and key. White blood cells respond to the antibodies. So they will be detecting the antigens. See, this is having a Y shape, and it goes and attaches to the triangular shape. Here you have the circular and triangular one. So it goes and fixes with the triangular structure present in the organ or in the region. So this is done with the help of plasma membrane. When this doesn't match, instead of this Y, if any other structure is there, it won't fix to that triangle. It is very specific for that shape. A triangle can come in and fix in this Y. The circle cannot go and fix in the Y. Okay, see, and this is another image. Immunoglobulin. See, it has shapes to get attached with this antigen, this antibody. It goes and fixes. They are activated. So it recognizes it is the thing present in our body. This is not the foreign thing. So likewise, it will be specifying antigen specificity. So with this, I conclude my lecture. Um, Thank you so much for your patient listening. And as usual, I acknowledge various uh, World Wide Web organizations for providing the GIF images for using in this PowerPoint presentation for making the students or the learners to get more information and better understanding. Other than this, I thank various authors for contributing their knowledge about the functions of plasma membrane. So, in the next video, I'll catch you up with the better information regarding a new title. Thank you all.